we need a title. Today, we're going to be talking about DNA structure. And anything that I write in my notes, you, as my pen breaks, should write in your notes.
DNA is twisted into two strands of a double helix. Each strand, is made of a sugar phosphate backbone connected in the middle by nitrogenous bases. And we're going to break each of these things down. That chord's super annoying, isn't it? Okay, maybe it's just annoying for me. So what I'm going to do is I'm going to draw my double helix here. So start with one strand like that. Go ahead and draw another strand like that. What I have currently drawn is the backbone. So the backbone is made up of alternating sugars and phosphates, and we're going to zoom in on that. But the backbone, the two strands here, are held together by nitrogenous bases, and the nitrogenous bases are in the middle. And I'm going to draw some long ones and some short ones because they actually are different sizes. So adding into my notes here that these are nitrogenous bases. And those are these things in the middle. So those are my nitrogenous bases. Feel free to tell me to speed up or slow down as we go. We're now going to zoom in on the backbone. So the backbone is made out of alternating sugars and phosphates. So first let's focus on one of those sugars. And in DNA, the sugar is called Deoxy Ribose. So it is a deoxy ribose sugar. All sugars end in ose at the end of their name. That's one of the ways that we remember that they are sugars. So deoxy ribose is a sugar similar to glucose, but it's not a glucose, it's a ribose. Deoxy means missing an oxygen, so it's a ribose without an oxygen, one fewer oxygen. And let's draw what this looks like. So this is one of those times where you might want to put your paper over your iPad so you can trace what I'm drawing. Or if you feel pretty confident in your drawing skills, go for it. We're going to start with an oxygen. And then deoxyribose is a 5-carbon sugar. So it's got 1, 2, 3, 4, 5 carbons. And off of some of these carbons, we have some OH groups. So off of the first carbon, we have an OH. Off of the third carbon, we have an OH. And off of the 5th carbon, we have an OH. And OH groups are connected by the oxygen to the carbon. So that is why, even though this is an OH group, we call it an OH group, I've written it HO, and that's because the oxygen is what is actually connected to this carbon. And if you remember back, like way back in the beginning, we learned that carbons have to have 4 bonds. So what I have not drawn here is there are hydrogens, hydrogens, to make it so that everything has the right number of bonds. Oxygen only needs 2 bonds, so these oxygens are all fine. This oxygen is fine. All right. One of the things that we have to do is we have to be able to talk about these carbon atoms in a way that makes a sense. When I say carbon 1, I need you to know which carbon I'm talking about. When I say carbon 4, I need you to know which carbon I talk about. So there's a naming technique that we do that involves counting the carbons. And when we count carbons, we always start at the oxygen and we work clockwise around the ring. So this is going to be carbon number 1. This one's going to be carbon number 2. Carbon number 3, carbon number 4, and carbon number 5. So when I count carbons, by starting at the oxygen, and counting clockwise. So count in the direction that the hands go around the clock. And this is going to be important because we're going to be talking about 5' and 3' ends of this carbon. DNA is read in a specific direction, and that is from 5' to 3'. And the way that we label this is the 5' end of the carbon. So 5' is the end of the sugar that has the 5 carbon, and the 3' is the end of the sugar that has the 3 carbon. So I've got my 5' carbon and I've got my 3' carbon. And that little tick mark there, that represents prime. So we call it 3' and 5'.
I'm a little confused.
Okay.
So when you're saying 5' carbon, are you saying that in total that's the complete of the sugar and the 3 is kind of like the quote unquote halfway mark, or like I'm just confused.
I literally mean the 3rd carbon.
Okay, so it just means that there's that many up to that point.
So when I look at any sugar, I can count the carbons. And so if I were to look at, say, glucose, what's not shown in this diagram, because I made this diagram for you when you didn't need to know about counting carbons, is that that's actually an oxygen. And then each of these points is a carbon. So this would be carbon number 1, 2, 3, 4, 5. And there's actually, in glucose, there's a 6th carbon up here. Again, I simplified this so you don't have it. But I could say that this is the 2' carbon. I could say this is the 4' carbon. I could say the 1' carbon. So in this case, I would say that when glucose connects to another glucose, they connect at carbons 1 and 4.
Oh, okay. Okay, that makes a lot more sense. Thank you.
You're welcome. That's a great question. The reason I'm doing that is because when we have our phosphates that we're going to add here, because the backbone is made up of alternating sugars and phosphates, the phosphates are going to connect to the 5 and the 3 carbons.
Okay, that makes a lot more sense. Thank you.
So let's add that now, because that's actually the very next thing. So part of the backbone is the phosphate. And a phosphate is a phosphorus, which is an atom, plus oxygens. So the way that this looks is I'm going to have my P, which is my phosphorus, and it's got the following groups attached to it. So it's got an OH at the top, a double bonded oxygen, an OH at the bottom, and an oxygen with a negative charge on the side. Miss Fitz just heard these notes, so she's probably getting very bored. She'll also be able to tell me all the little different things I do between classes because they're never quite the same. All right, so here's my phosphorus with my oxygens around it. I've got my sugar. These are going to connect through dehydration synthesis. So if you remember back when we did these things, how you lost an OH and an H, and that made water, and that allowed you to bind these things together. Well, the same is going to happen between my OH groups on my phosphorus and my sugar. So I've got an OH here and an OH here. What's going to happen is I'm going to lose an OH and an H, and then I'm going to bond these things together. So it's going to go carbon, oxygen, phosphorus, and that's going to join my phosphate and my sugar together. So let's see how that works. On the next page, I'm going to make my model. And I'm going to be using these little pieces that I'm going to tape in. So this is my sugar. It's a representation of it, but I can draw out, and you'll see that it has this peculiar shape that looks, I think it looks a little bit like a house with a chimney. So that's what I think of it as. But that is my sugar, and I'm going to tape my sugar onto my page, and then you are going to trace it. And about right there. Now, attached to my sugar, I have a phosphate. And my phosphate, I'm just going to draw the O's because the H's end up getting removed, but this one has a negative. Now my phosphate is going to be connected to my sugar, and it's going to be connected to my 5 sugar. So I'm going to put it up here. And then attached to this, I'm going to have my base. And in this case, and we'll spend some more time on bases, don't worry, I'm going to have my A base attached. And the bases attached to carbon number 1. So what I have here is what we call a nucleotide. A nucleotide has a phosphate, a sugar, and a base. And these are the monomers, and they float around in your cells. And then when you need to make more DNA, another nucleotide is going to come and attach to this. So I've got one nucleotide here, and another nucleotide is going to come in, and it's going to attach. Now, DNA is always built from 5' to 3'. You can actually only add things to this 3' carbon. And the reason for that is the molecule that makes DNA is called DNA polymerase, and it's an enzyme, which is a protein with a special job. And it can only hold this nucleotide so that this 3' end is exposed. The shape of that enzyme gives it this specific function, and it can only attach things right there. That's how its shape works. Imagine it like a screwdriver. You've got a flathead screwdriver and a Phillips head screwdriver. It has a certain shape. It can only do the job that it's given. So when I attach my next nucleotide here, I'm going to start by attaching a phosphate, and I'm going to attach this phosphate to carbon number 3. And so this is a P, O, double bonded O, single bond with a negative O, and there's actually a little line right there that connects my carbon to that oxygen. So now I have my phosphate, and I'm going to go ahead and I'm going to add another sugar. And then I'm going to keep building this backbone. I will add more bases, but not quite yet. So I'm just going to keep building. So next, I'm going to have another phosphate here, bonded to another sugar. draw one more. And then I'm going to give you a chance to catch up. Because as I said, I've done this about 1,000 times, so I can do it in my sleep. But you have not. So the backbone of DNA goes sugar, phosphate, sugar, phosphate, sugar. If I was to keep going, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And this is one strand of DNA.
Can you go back to the top? Sorry, I just like missed the A thing. Okay.
This is one strand of DNA. It's going to be attached to another strand of DNA that I have not drawn yet, and it's going to be attached through the bases. So the next thing we're going to do is actually take notes on what those bases are so that we can add the bases in.
What does the A thing mean?
It's a base. We're about to take notes on that.
Got it.
I know. It's almost like I anticipated that that would be your next question, Trey. Okay, so I'm going to leave this page open for my model notes, which means I'm going to go over to this side here. And I'm going to give this my title. So this page is going to be talking about my nitrogenous bases. And I know that today is kind of boring. We have a whole bunch of notes today, but I got to admit, I have been teaching this for like, I don't know, 11, 12 years, something like that. And if we just do all of it in one day, it will make so much more sense to you than trying to break it up. So we're just going for it today. And I promise it will come together. All right, nitrogenous bases. There are two different shapes that the bases might be in. So one of those shapes looks like this. I'm going to draw a six-sided shape connected to a five-sided shape. Now, in the past, anytime that there has been a point like this, we've said that there could be a carbon there. These are nitrogenous bases, but I am really lazy and I'm not going to draw all them out because there are like double bonds in here and a whole bunch of other stuff in here, and we will get to that later. But what you should know is at these points, it could be carbon or nitrogen, and there's actually a lot of nitrogen in these rings, which is why we call them nitrogenous bases. So instead of those points being all carbons, they are often going to be nitrogens in this case. Now there are two bases that have this shape, and those bases are called adenine and guanine. And for sake of ease, we're going to call adenine A and guanine G. The other shape is just a cute little six-sided shape. Now this nitrogenous base could be either thymine or cytosine, which we are going to call, you guessed it, T and C, because we're lazy. Now, these two rings together make a larger base than this single ring. Now, the bases have another set of names. And again, I'm not going to make you memorize this. That's why we're putting it all in your notes, because you can use your notes on all of the quizzes and tests and everything that we have in this class. You're allowed to use your notes. So know that I'm not going to make you memorize this, but I am going to tell you some tricks to help you memorize these things if that's something you're interested in. These ones here that are made up of two rings are called purines. And if it is made up of one ring, it's called a pyrimidine. Oops, there's the Y comes before the R. And here's how I remember this. I use a couple of techniques to help me remember this. Pyrimidine has a Y. Thymine and cytosine are bases that have Ys. Another way to remember this is that all gods are pure. So A and G are purines. My personal favorite way to remember this is because you'll often be looking at a model and you'll see that these ones are larger than these ones. Well, the word large has an A and a G in it. So the larger bases are A and G. And these cute little ones, the word cute has a C and a T in it. So the cute ones are C and T. Now that we have that, we need to know how these bond together. A bonds to T with 2 hydrogen bonds. A way to remember this is that there are 2 apples in the tree. 2 apples in the tree. G bonds to C with 3 hydrogen and my students told me that they can remember this because some very fancy people have a 3-car garage. And apparently C and G are fancy. Hydrogen bonds are weak. They're not very strong. They're an attractive force that works kind of like a magnet. So in the same way that magnets can be stuck together, broken apart, stuck together, broken apart, these bonds can easily break and reform. And that's going to become important later. But for right now, let's look at how these bonds work. So I'm going to draw one of these. If I draw this, there are only 2 options for what it is. What could it be?
Cytosine or thymine?
Yep, it could be C or T. Let's call this one T. If it's T, what's it going to bond with?
C.
Adenine.
A.
Adenine. So I'm going to draw an A here. 6-sided connected to 5-sided. And this is going to form 2 hydrogen bonds. So there's going to be a bond there and a bond there. Let's see how these other ones work. So I'm actually going to shift this over a bit because in my little model here, I can imagine, oops, that was just bad. It's not how I draw it. There we go. I can imagine that there's a backbone running right here and a backbone running right here. And so because you always have a big one paired up with a small one, it's going to keep those backbone strands the same distance apart. If you had a big one matched up with the big one, it would create a bulge in the strands. Or if you had a little one connected to a little one, it would make a pinch in the strands because they would literally be closer together because of their size. So this one I'm going to call G. G is connected to
C.
C, always. And how many bonds are there?
3.
3.
3. So I'm going to draw 3 hydrogen bonds. All right, we're doing so well. So let's go back to our model and let's add in our nitrogenous bases, and then we'll have to build this other side of the backbone. So I'm coming back over here. You tell me, if I've got A on this side, what am I going to have on this DNA strand?
T.
I'm going to have a T. And you'll notice with my shapes that I've drawn here, or rather used a computer to make, and then I printed them out. And my tape is sticking more to me than the paper. Not helpful. These fit together. Now, how many hydrogen bonds should I draw between my A and my T?
2.
2.
I should draw 2 hydrogen bonds in between there. Now, my nucleotide is made up of a phosphate, a sugar, and a base. The base is always connected to carbon number 1. So when I get my new sugar here, if I try and put it like this, let's count those carbons. So I've got oxygen, 1, 2, 3, 4, 5. This is carbon number 1. In order to make carbon number 1 attached to this base, I actually have to build this strand upside down. See how that works? I should have warned you about that before those of you that are tracing and copying put that in. My bad. So this strand is going to be built with the 5' carbon at the top, because this is carbon number 5. So the 5' carbon at the top and the 3' carbons are at the bottom. This strand, the 5' carbon is here, which means that this side is the 5' side, and this side is the 3' side, because that is carbon number 3. Now I'm going to flip this over because it's easier to write if I flip it over. And this is going to be my T. Now a nucleotide has a sugar, a base, and what am I missing right here?
Phosphate.
A phosphate. So I'm going to go ahead, I'm going to add a phosphate. And then this nucleotide is going to be complete. Now I said before that you can only, ooh, my computer's going to fall asleep. Let's see if I can figure out another way to plug it in. I just need more outlets on my computer. Let's see what this does to you guys. Did you, oh, am I back? Can you see my notes?
For me, it says no devices connected.
Okay, give me a sec. I was afraid of that happening. Stop share. Let's see here. Oh, yeah, shut that down. Okay, give me just a second. And I appreciate your patience. Are we back? All right.
Yeah.
Perfect. So before, what I told you is that you can only add to this 3' carbon, which means that you should be building the DNA in this direction. And that's going to be important for reading it, but this is a model and models are not always 100% right. So we're actually going to build it in the wrong direction, but I think it's more important that you understand how the structure works than how the building works. So attached to this phosphorus and this phosphate, I'm going to have another sugar. I'm going to label all these pieces. And you're going to get really good at drawing these. Now, if I have the chance to add these however I want, if this one is an A, this one's going to be a T. What one should I put right here? What do we think? I can pick any because it doesn't matter. Let's pick G. So if I have a G on this side, what has to be on the other side?
C.
A C. And if I have to have a C on that side, and they are connected to the 1' carbon, because these things are all specific shapes and sizes, you should see that they line up really nicely. How many bonds are between my G and my C?
3.
And I'm actually going to draw my C backwards because that's how we'll read it. So it will be upside down. All right, let's keep building. Connected to my sugar, I need to have my phosphate. And you'll notice that I keep drawing these minus signs. That is because DNA actually has a negative charge. Because the phosphates have this negative charge, and there are so many phosphates in the backbone, the overall charge of DNA is actually negative. Now, this phosphate is going to be connected to a sugar. And this is where my drawing starts to get a little bit bad. These should line up, and they do not line up because I drew this phosphate really big and this phosphate's really small. So just be aware of that. And then I'm going to draw another phosphate. And that phosphate's going to be connected to another sugar. Which is going to be connected to another phosphate. And you can't see that. So let's put in the rest of my nitrogenous bases. Let's go ahead and put a G on this side. G is connected to what?
C.
C. And how many hydrogen bonds should I draw between these?
3.
3. That worked out okay. So then let's have a T on this side. And an A. Can somebody tell me how I knew that this one is going to be a T and this one is going to be an A?
It's following a pattern.
It is following a pattern, but how can I tell just by looking at the shape? Which ones are large?
The big one's always the, if it's that little pointed like arrow-like thing, the big one's the A, the smaller one's the T. If it's the half circle, the big one's the G, the small one's the C.
Yep. That is true. That's how this works. All right, we are almost done. Almost done. So I'm going to flip this back over. Add a couple more things. So we labeled it before, but my 5' end is at the top, which means that my 3' end is going to be at the bottom. So the 5, you can't see this, the 5 carbon is here, the 3 carbon is here, which means that this is my 3 carbon, 1, 2, 3. This is going to be my 3' end. And this is important because DNA is always read from 5' to 3'. So when I'm giving somebody a sequence of DNA, I'm always going to go from 5' to 3'. So if I'm reading this sequence of DNA on this strand, I'm going to read it as A, C, G, T. Now let's look at this strand over here. Carbon number 1, 2, 3. What prime is this side? Is it 5' or 3'?
3'.
3'.
It is 3', which means that this end down here is going to be my 5' end, because this is carbon number 5. 1, 2, 3, 4, 5. Notice these go in opposite directions. What this means is that you also read them in opposite directions. So if somebody was asking me to read off this strand of DNA, I would read it off A, C, G, T. I would not read it T, G, C, A. That would be the wrong order, and it would end up making a different protein if that's how it's read. So we're always going to read from 5' to 3'. Couple more things that I'm going to add on to here is I need a whole nucleotide. So I'm going to put a box around one nucleotide. Notice that my nucleotide includes a sugar, a phosphate, and a base, and that the phosphate is on that 5' carbon. So here's another example of a nucleotide. Sugar, phosphate, and nitrogenous base. These make up a nucleotide. A little more labeling. We've got some hydrogen bonds. And so a strand of DNA is made up of nucleotides. Are we okay with this so far? Let's take our final notes. So I'm back on this page. The last thing that I'm going to take notes on is a nucleotide. So first of all, DNA is a polymer. And polymers are made of monomers. Well, the monomer of DNA is a nucleotide. So last drawing that we're going to do is we're going to draw a nucleotide. I've got my phosphate bonded to my sugar. Oops. Bonded to my sugar. It's got to have 5 carbons in it, so you have to build it correctly. Bonded to my nitrogenous base. So this is called a phosphate. This thing over here is my nitrogenous base. And this is my sugar, which is also known as deoxy ribose. And DNA is a nucleic acid. It's called DNA because it's deoxyribonucleic acid. I'm going to use some markers to color this. So let's make my phosphate red. I'm going to make my sugar blue. And you'll notice that these shapes should look somewhat similar to what we made on our model. Notice that the sugar is like the shape of a house with a chimney. And then my nitrogenous base, well, those comes in all different shapes and sizes, so those are a little bit harder to model out. But there we go. And those are our notes.